Welcome to a deep dive. You ready to unlock some secrets of molecules? Really complex ones.
Sounds exciting.
We've got some awesome sources this time.
Oh, yeah.
We're going to be working with Dorothy Hodgkin. You familiar with her? Nobel lecture from 1964.
Okay, yeah, I've heard of her. That's going way back.
It is the early days.
Yeah.
Then we'll jump to 2009. Venki Ramakrishnan.
Oh, wow. Okay.
Excerpts from a lecture and some slides. He tackled the ribosome.
So,
That's a big one.
huge. To round things out, we've got a 1965 lecture by Robert Burns Woodward.
The master.
Oh, yeah, chemical synthesis.
Hmm.
It's amazing to me how
Yeah.
these pioneers, each working in their own time,
Right.
they help build this understanding of
Yeah.
molecular structure.
And just pushing the boundaries of what was possible right at that time.
Absolutely. We're going to see how science evolves and
Yeah.
builds upon itself.
Hmm.
So let's start with Hodgkin. Her journey began with a simple spark of fascination,
Okay.
a children's book by W.H. Bragg.
Ignited her passion for visualizing molecules.
Wow, I love that. That's amazing.
Talk about finding your calling in unexpected places.
Really does. Speaks to that power of, clear, engaging science writing.
Yeah.
Even for these really complex topics. I mean, who
Mhm.
knows how many, future scientists were inspired by that book.
It's true. And it led her to some incredible discoveries.
Yeah.
She started with simpler molecules, but eventually found herself at Cambridge working with J.D. Bernal.
Oh, wow.
His lab was like a hub. All sorts of research,
Yeah.
digestive enzymes, pepsin,
Oh, wow.
speculating on how muscles contract, like
Oh, wow. That's amazing.
all sorts of things.
That's a fascinating lab. Bringing together people from all these different areas
Yeah.
to really tackle these big questions must have been an incredible environment.
Sounds like a scientific playground.
Yeah.
Right? And it was there that Hodgkin truly mastered X-ray crystallography.
Okay.
So for anyone who's unfamiliar with this technique,
Right.
imagine shining a super powerful light through a crystal
Okay.
where light scatters.
Yeah.
And by analyzing that pattern,
Right.
you can figure out the arrangement of atoms within the molecule.
It's like trying to figure out the shape of something just by looking at its shadow,
Yes.
but obviously much more complex.
Much more complex.
Yeah.
To make sense of these patterns, you need to understand electron density.
Right.
So this is essentially a map. It shows the distribution of electrons within the molecule.
Yep.
Heavier atoms with more electrons
Yeah.
act as stronger signals. They help us pinpoint their position and piece together the structure.
So it's like landmarks on a map.
Yeah.
Guiding us towards the full picture.
I like that.
Yeah.
And all of this without computers.
Right.
Hodgkin was working with tools like Patterson synthesis,
Yeah.
beavers,
Yeah.
and lips and strips, literally strips of paper
Love
with
it.
calculations on them.
I can't even imagine.
So patience.
Yeah. It's mind-boggling. Like, what we can do in hours now would have taken them months
It's incredible.
of painstaking calculation.
Talk about dedication.
Mm.
And it led to some groundbreaking discoveries.
Yeah.
Cholesterol iodide. This was key in confirming the structure of sterols,
Right.
which are essential components of cell membranes. So that means, for you, this is a better understanding of how your cells function at the most basic level.
Yeah.
Then there's the wartime challenge. Penicillin. Tiny amount to work with.
Oh, wow.
The pressure was on to determine its structure.
That must have been intense.
I know. Did they succeed?
They did. That was huge.
Yeah.
Paved the way for understanding how it works. Revolution in treating bacterial infections. I mean, think about the impact.
Countless lives.
Countless lives.
It's incredible
Yeah.
how fundamental these discoveries were.
Yeah.
And she didn't stop there. Vitamin B12, even more complex.
Hmm.
And can you imagine the surprise when they finally uncovered the structure?
Oh, yeah.
It was unlike anything they'd seen before. This intricate three-dimensional puzzle.
That was a massive leap forward
Yeah.
and required collaboration with, people who had access to those early computers. It shows how technology plays a role in this kind of scientific progress.
It was a team effort for sure. Speaking of complex molecules,
Yeah.
we can't forget about cephalosporin C, another antibiotic with a fascinating structure.
Okay.
It actually has a four-membered ring,
Huh.
quite unusual in nature.
Yeah.
And it originates from a similar source to penicillin,
Okay.
but in an oxidized form.
Yeah.
It really highlights how nature just builds on these similar blueprints
Yeah.
to create diversity.
It's like evolution at the molecular level, right?
It is.
There's always something new to discover, even in seemingly familiar places.
Now let's dive into one of Hodgkin's most ingenious techniques,
Okay.
anomalous dispersion.
Okay.
Used it to study cobiuric acid.
A precursor to vitamin B12.
Okay.
But before we get too deep,
Yeah.
how would you explain anomalous dispersion?
Oh.
In simple terms.
Okay, so think about it this way. Certain atoms, like the cobalt in cobiuric acid,
Okay.
can change the phase of the X-rays that
Okay.
are passing through them.
And by carefully measuring these phase changes,
Yeah.
we can pinpoint their location within the molecule.
So it's like those atoms are giving us extra clues.
Yeah. It's like they're leaving these unique fingerprints on the X-ray pattern.
That's pretty cool. Didn't they see this effect visually in the data?
He did. You can see it. Like this asymmetry in the X-ray reflections from the crystals.
Wow.
It's direct evidence of anomalous dispersion in action.
Like seeing the invisible world come into focus?
Yeah.
What's even more amazing, the electron density map they created.
Yeah.
So clear.
Really?
That they could see not just the chemical formula, but even the hydrogen atoms.
Wow. Talk about a high-resolution image. That's incredible.
Right.
Yeah.
And, just to double check,
Yeah.
they compared their structure to a synthetic nickel-corin derivative,
Oh, okay.
made by Dunitz.
Okay.
Further confirming.
Multiple lines of evidence.
Exactly.
It's like having multiple witnesses corroborating the story.
That's right.
Okay, so they conquered cobiuric acid.
But what about B12 itself? The coenzyme
Right.
plays a critical role in all these processes,
Mm-hmm.
but it was notoriously difficult to isolate, unstable, real challenge to study.
This is where X-ray crystallography really shines.
Yeah.
Allowed them to see something that nobody had anticipated. A direct link between the cobalt atom
Okay.
and the adenosyl residue.
Wow.
So this unexpected connection, it was a major aha moment.
So it wasn't just about the structure, it was about understanding how the pieces fit together.
That's right. That's right. This discovery really explained why it was so unstable, and that intricate geometry, it's key to understanding its function,
Yeah.
particularly in transferring those methyl groups. It's like,
Form follows function.
Yeah.
Makes a lot of sense. But even with the structure, so many questions.
oh yeah,
Especially how it works within a living cell, right?
right, right. We're talking about a process that's constantly in motion. X-ray crystallography, it gives us this snapshot. But to really understand the dance, we need to see them in action within the enzymes.
Right.
Yeah.
So it's like having the still photo versus the full-length movie.
Exactly.
Both valuable,
Mhm.
but different parts of the story.
Totally.
And this brings us to Hodgkin's work on an even larger and more challenging molecule,
little.
insulin, key to regulating blood sugar.
Yeah.
We knew the chemical structure from Sanger's work,
Right.
but its exact mechanism, still a mystery. Even today, despite all this research,
True.
still so much we don't understand.
That's the beauty of science, right?
Yeah,
Always more to explore.
always more to explore. So Hodgkin discovered that it could actually crystallize in different forms.
Oh, wow.
And these forms are related to its function.
Interesting.
So for example, it's dimeric,
Mhm.
meaning two molecules bound together
Right.
in acid salts.
Yeah.
But it potentially forms these larger aggregates
Okay.
in its cubic form.
Interesting.
And definitely when zinc is present. So the environment influences the shape,
Right.
and the shape influences how it works.
That's fascinating.
Isn't that interesting?
Like-
And there's a historical connection here.
Oh.
Back in 1927,
Oh, wow.
Abel actually observed cubic insulin crystals
That's amazing.
decades before.
Yeah.
So even early observations can lay the groundwork for later discoveries.
It's true.
It's like a scientific echo across the years. But insulin presented new challenges.
Oh, yeah.
Introducing those heavy atoms
Right.
proved tricky.
Oh.
It was a real marathon,
Yeah.
full of obstacles, exploring all sorts of techniques,
I can imagine.
anomalous absorption effects
Oh.
to try and crack the code.
Okay, so for those who don't know, anomalous absorption effect.
Or how would you explain that?
So basically, when X-rays hit certain atoms,
Right.
they can be absorbed in a way
Yeah.
that changes the intensity of the scattered beams.
Huh.
And, by analyzing those changes,
Yeah.
you can glean more information about the positions of those atoms.
It's adding like another layer of detail
Exactly.
to our map.
Yeah.
It's incredible how scientists find these subtle clues to piece together the puzzle.
It's amazing.
But I feel like we've been talking about Hodgkin for a while.
We have.
You ready to move on to Venki Ramakrishnan and the ribosome?
Let's do it.
Okay, buckle up. We're about to get molecular.
All right. The ribosome, it's like this molecular factory, It takes the instructions from your DNA and churns out proteins based on those blueprints.
It's a pretty important job.
Huge. It's a complex machine. 2 main subunits. And scientists have been working for years to try and unravel all its secrets.
It's a big one to tackle, for sure.
Yeah.
So Ramakrishnan, he started by focusing on the structures of individual ribosomal proteins.
Right.
Kind of like the building blocks of this machine.
Yeah.
But he realized that to really understand how it works, he needed to see the bigger picture.
Right.
He needed the structures of the entire subunits.
Like trying to figure out how a car works by looking at the engine, the wheels, seats separately. You got to see how it all comes together.
You need the whole system.
Yeah.
But crystallizing ribosomes,
Right.
not easy.
No.
It's a large structure,
Mm.
fragile,
Oh,
difficult to work
they're a
with.
real headache for a crystallographer.
How did he even manage to get those high-resolution structures?
This is where his ingenuity comes in. He made some real landmark achievements in obtaining, diffracting crystals, and his innovative use of a technique called multi-wavelength anomalous diffraction, or MADI for short.
Mediphasing.
Yeah.
Break it down for us non-crystallographers.
Okay, so imagine taking multiple X-ray pictures of the same crystal,
Okay.
but each picture is taken with a slightly different wavelength of light.
Okay.
So by analyzing those subtle differences in those images, you can pinpoint the locations of specific atoms.
Okay.
It's like using different colored lights to highlight different parts of a stage.
I see. It's about exploiting those subtle differences.
Yeah,
To
exactly.
extract more information.
Yeah,
Didn't he face some initial roadblocks with MD-phasing, though?
he did. And this is where the story gets really interesting.
Yeah.
Initially, he calculated that there wasn't enough methionine in the 30S subunit for MADI phasing to really work.
Oh, so what did he do? Just give up?
No, he had to think outside the box a little bit,
Yeah.
find alternative atoms that would work.
Okay.
He ended up using lanthanides,
Interesting.
which is a group of rare earth elements,
Okay.
and osmium hexamen. It was a real challenge. Classic example of scientific problem solving,
Yeah.
adapting your approach when you face a challenge.
It's inspiring to see how a setback can lead to a more creative solution.
It can.
So besides atom phasing, are there other methods for determining the phases?
Yeah. There are a couple of other ones.
Okay.
Heavy atom clusters, molecular replacement.
Go ahead.
Heavy atom clusters. Think of it like attaching little tiny weights to specific parts of the molecule so they kind of stand out in that X-ray data.
Okay.
And then molecular replacement uses a previously solved structure
Okay.
as a starting point to help solve the new one.
So it's like having a reference guide
Exactly.
to help you solve a puzzle.
Yeah.
So a whole toolkit of approaches to tackle these complex structures.
It's all about finding the right tool for the job.
Absolutely.
Yeah.
Let's dive into what they discovered about the 30S subunit.
Okay.
What did this structure reveal about the inner workings of the ribosome?
This was a major moment in ribosome research.
Okay.
30S subunit structure
Yeah.
revealed a wealth of information.
Okay.
Some of it was really unexpected.
Like what?
Well, they could clearly see the right-handed double helical structure of the RNA within the subunit.
RNA, not DNA.
RNA, crucial player,
Interesting.
confirmed its important structural role.
Very cool.
And they were able to identify specific elements of the structure, helix 44, and figure out where those ribosomal proteins fit into the electron density.
So they're putting together this three-dimensional jigsaw puzzle.
Exactly.
Anything else surprising?
They did, and this was a major shift in our understanding of the ribosome. They discovered that the functional sites,
Right.
where the real action happens, are primarily composed of RNA,
Okay.
not proteins. And the interface between the two subunits, also dominated by RNA. This challenged the view at the time that proteins were the main players.
Wow. So RNA taking center stage?
RNA is the star.
Not just the supporting actor.
Not at all.
That's a big deal.
It was a game changer.
Yeah.
Of course, proteins still play a role,
Yeah.
but this highlighted RNA's significance.
Okay.
They also found that many of the proteins have these long tails that interact with the RNA.
Okay.
It's this intricate interplay.
So it's a delicate dance between RNA and proteins working together
That's a good way to put it.
to build the molecules of life.
Yeah.
Now, the story of the 30S subunit doesn't end there, right?
No, it doesn't.
This structure helped us understand how antibiotics work.
Yes. 30S subunit opened up this new avenue for studying how antibiotics target bacteria.
So it's like having a blueprint of the battleground.
It's-
showing us where those molecular weapons strike.
Yeah,
So they were able to see where the antibiotics bind.
exactly. They analyzed something called difference Fourier maps,
Okay.
which highlight the changes in the electron density when that antibiotic is present.
So you're comparing before and after.
Yeah.
to see where it latches on.
You got it.
And one of their remarkable findings,
Yeah.
the 30S subunit with 3 different antibiotics bound simultaneously.
3.
Streptomycin, spictinomycin, and paromomycin.
Wow. Caught it in the middle of a molecular siege.
What do these structures tell us?
They provided a ton of information about how they interfere with bacterial protein synthesis.
Okay.
Streptomycin distorts the ribosome, leading to errors in decoding.
Okay.
Spectinomycin blocks the movement of tRNA, and then paromomycin,
Yeah.
which we'll discuss a little later, gives us incredible insights into this process of decoding itself.
Decoding. You mean how the ribosome accurately translates the genetic code, making sure the right amino acids are linked together to build the proteins?
It's crucial for making sure the right protein is made, like making sure you follow the recipe correctly.
You mix up the ingredients, you get a different dish.
Exactly.
So what's the challenge with decoding? How does it ensure it's selecting the right tRNA?
So it's a remarkable process.
Yeah.
And it starts with the genetic code itself. Ever since we cracked the code, we've known that it's degenerate.
Degenerate.
Meaning multiple codons can code for the same amino acid.
Oh, so there's redundancy built in.
Exactly. And because there are fewer tRNAs than codons,
Right.
Crick proposed the wobble hypothesis.
The wobble hypothesis.
Yes.
Doesn't it mean that some mismatches are tolerated at the 3rd position of the codon?
You got it. But here's the thing. The energy difference between a correct base pair and an incorrect one isn't really enough to explain the accuracy. The ribosome gets it right almost every time.
So there must be something more to it.
There is.
It can't just blindly accept any tRNA that comes along.
No. Scientists began to suspect that the ribosome itself plays an active role.
It's like a quality control inspector in that molecular factory.
Okay.
So early evidence for this came from those studies with streptomycin.
Okay.
Binds to the 30S subunit
Yeah.
and increases errors in protein synthesis.
So by messing with the ribosome, it throws a wrench in the decoding process.
Exactly. And these findings led to this idea of a decoding center
Okay.
within the ribosome,
Right.
where that pairing is carefully inspected.
So a proofreading mechanism.
That's the idea. But then the discovery of something called a Hirsch suppressor, this fidelity mutation.
Okay.
It's located far away from the tRNA in acodon. Challenged this view.
So a mutation far away affects the accuracy.
It does. And this led to an alternative view called kinetic proofreading.
Kinetic proofreading?
Yeah.
What's that?
In this model, the ribosome isn't directly inspecting the pairing.
Okay.
It's more about modulating the rates of various steps.
Okay.
It's more about controlling the timing of events
Okay.
than physically checking the base pairs. So like a series of hurdles.
Yeah.
And incorrect tRNA is more likely to trip over.
Exactly. So
Right.
incorrect tRNAs are more likely to dissociate before or after a key step,
Well,
involving a protein factor called EF2 and the energy molecule GTP.
so controlling the flow of events.
That's it.
Okay. But if it's not directly inspecting, how can a mutation so far away affect accuracy?
It's a great question.
Yeah.
And it really highlights this interplay between structure and function.
Right.
Kinetic proofreading suggests that both the ribosome and the tRNA are somewhat passive,
Okay.
players
Get
in
to
this
that.
process.
Okay.
Mutations, even those far away, can influence the rate of GTP hydrolysis by EF2.
Okay.
That's a key step
Yeah.
in this proofreading process.
Got it. So a delicate balance of speed and accuracy.
Exactly. But the story doesn't end there.
Okay.
As scientists dug deeper, they realized that original view of the decoding center might not be entirely wrong.
So more to the story. It does actually inspect them directly.
It appears so. And the structure of the 30S subunit, along with those studies using paromomycin, provided some clues.
Paromomycin, the one that was bound to the ribosome along with those other two.
That's the one. The structure of the 30S subunit bound to paromomycin showed something really remarkable.
Okay.
The antibiotic stabilizes the conformation of 2 key bases,
Okay.
A1492 and A1493.
Okay.
In a way that suggests they can directly inspect that minor groove of the codon-anticodon helix.
So they're like molecular calipers checking for the right fit.
Exactly.
So it does have a way of double checking,
It does.
not just relying on the timing.
Again.
Okay.
These 2 bases, along with another one called G530, seem to work together
Okay.
to sense the precise geometry of those base pairs.
Didn't you say it's super difficult to crystallize ribosomes with mRNA?
It is. But Ramakrishnan and his team made this really fortunate discovery.
Okay.
They realized that the A site of the 30S subunit, where decoding occurs,
Right.
is exposed to these large solvent channels in the crystal.
So there's a way to sneak in the ingredients through a back door.
You got it.
Okay.
Allowed them to soak small RNA molecules that mimic mRNA and tRNA directly into the crystals.
So they set up this controlled environment
Yeah,
to observe the interactions.
that's exactly right.
I love it.
Yeah.
What did they learn from these experiments?
Okay, so their findings were really groundbreaking. They showed that when a cognate tRNA,
Cognate meaning a perfect match?
perfect match, binds to the 30S subunit,
Yeah.
it causes a change in the conformation of those three key bases,
Okay.
A1492, A1493, and G530.
Right.
So these bases, they fit into that minor groove of the codon-anticodon helix, but only if the base pairing is correct at those first two positions.
So they're checking for the right handshake.
Yeah.
Before allowing the tRNA to proceed.
Exactly.
I love it. This interaction provides a beautiful explanation for the wobble hypothesis.
It does.
Being strict about the first two positions.
Exactly.
More lenient at the wobble position.
That's right.
It's all about that balance between accuracy and flexibility.
Yeah.
But you also mentioned global conformational changes.
Right. So they discovered that cognate tRNA binding doesn't just cause those local changes in the decoding center.
Right.
It triggers this large-scale conformational change in the 30S subunit itself.
So it's not just about checking the code, it's about locking the correct tRNA into place.
Precisely.
Okay.
And this domain closure,
Yeah.
as we call it,
Okay.
only happens with a cognate tRNA unless paramomycin is present.
Paramomycin again.
It's a key player in this story.
I love it.
So this finding provided a really crucial link between structure and function.
Okay.
Kinetic studies had shown that cognate tRNAs accelerate the rate of GTP hydrolysis by EF2.
Right.
Paramomycin mimics this effect even with near-cognate tRNAs.
Near cognate. What does that mean?
So a near-cognate tRNA is one that has a single mismatch
Okay.
with the codon.
So almost a perfect match.
Almost. And this is where paramomycin throws a wrench into things
Okay.
by stabilizing the ribosome. Yeah. In its closed conformation, even with that near-cognate tRNA, it allows the wrong amino acid to be added.
So it's like tricking the ribosome into accepting an imposter.
Exactly. And this highlights the importance of that domain closure for GTP hydrolysis.
Right, because that's the point of no return.
It is.
Once that happens.
The tRNA is committed. Yeah.
But why is domain closure so important for GTP hydrolysis?
To understand that, we need to zoom out.
Okay.
Look at the whole ribosome,
Yeah.
not just the 30S subunit.
Okay.
This is where cryo-EM comes into play.
Cryo-EM, the incredibly detailed 3D images.
You got it.
Yeah.
Cryo-EM studies had shown that the tRNA, when bound to the ribosome as part of this complex with EF2, is distorted. Bent out of shape.
Huh. So it's not just sitting there passively.
Not at all. So based on this observation, they proposed a model
Okay.
where the energy from that correct codon-anticodon pairing,
The handshake.
that perfect handshake,
Yeah.
helps to drive the domain closure. And this closure stabilizes the distorted tRNA.
Okay.
And this distortion promotes GTP hydrolysis by EF2.
So it's a chain reaction.
Yes.
tRNA binds,
Yeah.
ribosome closes, tRNA distorts
Yeah.
GTP hydrolysis.
That's it. Like a carefully choreographed molecular dance.
It's amazing.
Yeah.
And it explains why those mutations in the ribosome and tRNA can affect fidelity,
Exactly.
because they can make it easier or harder to achieve that domain closure,
Right.
throwing off the timing.
Exactly.
Wow. These tiny molecular tweaks can have a huge impact.
They can.
How do they even get a structure of the whole ribosome with EF2 bound?
Huge challenge. Took years to overcome.
That's
Remember
it.
that protein L9?
The one they had to delete to study the 30S subunit?
That's the one. Well, they figured out how to get rid of it, or
Okay.
at least the part that was causing problems.
Okay.
They used some genetic techniques to just delete the gene.
So they did some molecular surgery,
Exactly.
removed the troublesome bit,
Got it.
but kept the rest functional.
That's right. But here's another twist.
Okay.
Deleting that part of L9 actually prevented the ribosome from crystallizing.
Yeah.
Back to square one, had to start screening all over again.
Wow.
And this is where the story gets even more interesting.
Okay.
Years earlier, they had found some promising conditions for an EF2 complex, but they abandoned them.
They abandoned them. Why?
Well, the scientists working on that project left the lab. Research was put on hold.
Wow.
Really shows you how science is a collaborative effort, and sometimes progress depends on the right people being in the right place at the right time.
It's true.
Yeah.
So those crystallization conditions just disappeared into a lab notebook somewhere.
Thankfully, no. Years later, they stumbled upon a new crystal form of an EFG complex.
Okay.
Another GT-PACE factor, using conditions that were strikingly similar to those initial hits for the EF2 complex.
Oh, wow. What are the chances?
So they resurrected those old conditions,
Okay.
and they got crystals of the EF2 complex bound to the ribosome.
They did.
Yeah, but they had to use one more trick.
Okay.
They added a molecule called curamycin,
Curamycin.
which stalls the complex after GTP hydrolysis.
Okay.
It's like hitting the pause button,
Yeah.
allowing them to capture that snapshot.
So curamycin, another antibiotic.
Yeah.
These molecules are really playing a starring role in unraveling these mysteries.
They are. And the structure they obtained with curamycin was really amazing, confirmed many of their hypotheses about the decoding pathway, and gave us a much deeper understanding of how it all works.
Okay, I'm on the edge of my seat. Tell me, what did this structure reveal? How does it all come together?
It really is a combination of years of research, confirms that sequence of events we've been discussing, recognizing the right codon, GTP hydrolysis, all of that. It all starts with the tRNA escorted to the ribosome by EF2,
Okay.
which is bound to GTP.
So EF2 is like a chaperone.
Yeah, making sure it gets to the right place.
Okay.
Once it arrives, the tRNA starts sampling different codons on the mRNA, kind of like trying on different shoes until it finds the perfect fit.
And if it's the right fit, that decoding center springs into action.
Yes.
Double checking everything's in order.
Those three bases, A1492, A1493, G530, they interact with the minor groove, but only if the base pairing is right at those first two positions.
It's a molecular handshake.
I like that. And that recognition triggers the 30S subunit to close up, bringing everything closer to EF2.
Okay.
Causes all these tiny little shifts in the structure, ultimately leading to a key interaction between the tRNA and EF2 being broken.
Like a chain reaction.
Yeah.
One change setting off another.
Exactly. And that's when the GTP molecule becomes exposed, making it vulnerable to hydrolysis. GTP hydrolysis, the point of no return.
Right.
Once that happens, EF2 changes shape, releasing the tRNA and the ribosome.
So the tRNA is finally free to move to the peptidyltransferase center.
Yes.
Add its amino acid.
That's
To
right.
the growing protein chain.
Exactly.
It's like it's passed all the tests.
Yeah.
And it's ready to contribute.
It's a beautiful system. And this structure with EF2 and curamycin allows us to really see these steps in such detail.
Amazing.
Yeah. Confirms what we thought we knew about decoding.
Power of X-ray crystallography.
It is. But remember, we're not just talking about structures.
Right. Function.
We're talking about function, the dynamic processes that make life possible.
Not just a static picture, right? It's about understanding how they move and interact, how they work together to carry out these essential tasks.
Exactly. Okay, I think we've explored Hodgkin's world
Yeah.
and Ramakrishnan's world.
Ready for a change of pace?
I think so. Let's talk about Woodward.
All right. Robert Burns Woodward,
A legend.
chemical synthesis.
He wasn't just satisfied with observing molecules.
Okay.
He wanted to build them, create them from scratch.
So we're moving from observing to building.
We are. And we're going to focus on his work with cephalosporin C. Remember, we talked about it earlier.
Right. Antibiotic related to penicillin.
Yes. With that beta-lactam ring, Woodward took on this challenge that most people thought was impossible.
Really?
He decided to synthesize it from scratch, presented his unpublished work on it at his Nobel lecture in 1965.
What? Talk about confidence.
yeah.
What made it so difficult? I mean, wasn't it similar enough to penicillin that they could have just used existing methods?
Well, it does share some features with penicillin, including that beta-lactam ring, which is really fragile.
Yeah.
But with cephalosporin C, you need to make sure all the atoms are arranged in a very specific way in three-dimensional space.
Oh, I see.
It's called stereospecific synthesis.
Okay.
And it's not easy.
It's like building a house of cards.
Yeah,
But each card has to be perfectly placed.
exactly. To make things even more challenging.
Yeah.
He chose to start with L plus cysteine,
Okay.
an amino acid,
Right.
that already contains a chunk of that structure.
So you're starting the puzzle with a few key pieces in place.
Yeah,
But still tons of work to do.
tons of work. But cysteine is a bit of a wild card. It's got all these reactive groups that could cause all sorts of problems,
Yeah.
unwanted side reactions.
Right.
So they had to really carefully protect those groups. It's like putting molecular safety goggles on them.
I love it. So they could manipulate it without causing chaos.
That's right.
So they were able to control its reactivity.
They were. It was impressive.
Yeah.
They also had to introduce a nitrogen atom into a part of the molecule that's resistant to attack.
Right, into that methylene
You're
group.
remembering the details. I try.
But they came up with this novel reaction using dimethyl azotic carboxylate.
Okay.
This allowed them to put that nitrogen atom exactly where they wanted it,
Wow.
in the correct orientation.
So they threaded the needle
They did,
with pinpoint accuracy.
while navigating a minefield of potential side reactions.
It's a testament to their mastery.
It is.
Okay, so they got the nitrogen in place.
Hmm.
What's next? Building the beta-lactam ring.
That's right. And that ring is so fragile.
Yeah. Prone to falling apart.
Right.
They had to handle it with molecular tweezers,
I can imagine.
carefully converting the molecules step by step
Yeah.
until they finally formed the beta-lactam ring.
I bet there was tension in the lab when they were working on that stuff.
Sure.
Did they confirm the structure?
Of course, they used X-ray crystallography
Of course.
to confirm the structure of the beta-lactam.
Smart.
Yeah. And then came the final stages of the synthesis, adding more pieces, building out that core structure.
So building the foundation, then the walls, then the roof.
Exactly. And some clever tricks along the way. For
Yeah.
example, using a protecting group that could be removed without damaging the beta-lactam ring.
Like a molecular parachute.
Yes,
So they could land safely at the destination.
exactly. And after a series of carefully choreographed steps,
Mm-hmm.
they reached their goal.
Synthetic cephalosporin C.
They did.
They compared it with a natural compound.
They did perfect match. Even tested the antibacterial activity to be extra sure.
Wow. That is a remarkable story of scientific ingenuity.
It is,
Perseverance
for sure.
really makes you appreciate the complexity
Yeah.
and the effort it takes to not just understand, but recreate them.
It shows you we can not only understand these intricate structures, but also become molecular architects ourselves.
That's a great way to put it. This has been quite the journey.
It has.
X-ray crystallography,
Yeah.
chemical synthesis, seeing how they've revolutionized our understanding of molecules.
It's incredible.
From the smallest building blocks to the complex machinery of life,
The challenges, the breakthroughs.
the collaborative spirit,
It really is a testament to human curiosity.
and the pursuit of knowledge.
Absolutely.
It leaves us with a sense of wonder, doesn't it?
It really does.
So many secrets waiting to be unlocked. Who knows what we'll explore next?
What challenges we'll overcome?
I'm excited to find out.
Me too. And, maybe you'll be the one to make the next big discovery.
You never know.
Keep diving deep. Keep asking those questions. Never lose that sense of wonder.
That's a great note to end on. Thanks for joining us on this deep dive.
Thanks for having me.
And we'll see you next time for another exploration of the fascinating world of science.